just as a bit of background, so patients who are either ineligible or unwilling to undergo potentially curative therapy for diffuse large B-cell lymphoma, so relapsed refractory disease, who are not candidates for high-dose chemotherapy with autologous stem cell rescue or for CAR T therapy, have a poor prognosis. Now, the prognosis has certainly changed somewhat over time. There are some available therapies now that we didn't have even a few years ago. So, for example, we've added Naratuximab to our treatment regimen, which is an anti-CD79 ADC. And then we've also had selinexor, an exportin-1 inhibitor, tafasitamab, which is anti-CD19 monoclonal antibody, along with lenalidomide, has been studied in that same population. And we've recently added lancastuximab tesirine, an anti-CD19 antibody-drug conjugate. But again, none of these are, thought to be curative therapies. And clearly, there is an additional unmet need for patients in that space. There's also a need for novel targets. And, one such target that certainly seems to be a very reasonable one to pursue, especially based on the results of this abstract, is CD37. So CD37 has near ubiquitous expression in a variety of non-Hodgkin's lymphoma, especially diffuse large B-cell lymphoma. So it's around 90% expression in diffuse large B-cell lymphoma. And one of the things that makes it such a good candidate for a potential antibody-drug conjugate is the fact that this is an internalizable cell surface marker. So, the marker we obviously associate most with diffuse large B-cell lymphoma is CD20, but CD20 does not internalize very well. However, CD37 does, and the whole goal of giving ADCs is to preferentially get your payload, which in this case is an antitubulin payload, directly into the cell. So an internalizable cell surface receptor is obviously what you're trying to target. Now, what they found with preclinical testing is that if they co-administer rituximab, an anti-CD20 monoclonal antibody, with this anti-CD37 ADC, that you'll actually get more internalization of CD37, therefore more payload delivery into your target cancer cells. So this was a study taking a look at Naratuximab Emtansine in the treatment of various non-Hodgkin's lymphomas. And initially, they looked at all comers. So we had, aggressive lymphomas like diffuse large B-cell lymphoma, as well as, indolent lymphomas like follicular, marginal zone, and, everything in between like mantle cell. And in the phase 2 study, in the phase 2 portion, they focused then on just diffuse large B-cell lymphoma. I'm sorry, not in the phase 2. In the second portion of the trial, they looked at just diffuse large B-cell lymphoma in 2 different dosing cohorts. One was a Q week and one was a Q3 week. And what they found was several things that I think are notable. So the first was that this was a very effective therapy. So response rates, meaning PRs or better, was almost half of the patients. And of the responders, many of the responses were deep and durable. So most of the responders actually achieved a complete remission. And of the people who are responding, the median duration of response had not yet been reached at 12 months of follow-up. Furthermore, the therapy was also quite well tolerated. So despite the fact that these were not the hardiest of patients, less than 1 in 10 patients actually had to discontinue treatment due to an adverse event. One of the concerns that we have with ADCs is neuropathy. We also have concerns of LFT abnormalities, and we didn't see any of that in this particular study. So, you have a therapy that's, obviously very effective, targeting an unmet need population, and it's also very well tolerated. So when you think about, all of these things combined, you can see that this would lend itself very well to future studies with other, targeted combinations in this particular space that may lead to even, more durable responses, hopefully a cure.